So session 2 is vaccine and immunotherapy. I'm Dr. Jean-Pierre Routy from McGill University in Montreal. Definitively a winter in this Brisbane is a bit different than in other parts of the world. So we're very happy for them. I am also with Gabriela Turk from Buenos Aires, co-chairing this session. And we ask first all the speakers to come on stage and take their seats, and they can find their name by their seats. So let's start. And the first speaker is from Barcelona. So Beatriz Mose Pujadas will present on HIV ACAT T-cell immunogen and clinical trials.
So thanks everyone for being here and the organizers for inviting me to give this talk. So these are my conflicts of interest. And I hope you leave this session or leave this talk. I would give just an important message that is, I think there are therapeutic vaccines that are somehow effective in controlling viral load, and they should be part of the combinational trials, and I hope I can convince you about that. So we know from the therapeutic vaccines candidates expressing HIV entire proteins have shown limited activity so far in randomized controlled trials, both in modulating the T-cell response and the ability to contain viral replication. And here you have a picture of the, I would say, the most advanced T-cell immunogens in the field. We have what we call the full proteins design. That would be basically they regularly express GAG, POL. Some of them, they also express envelope proteins. And I think there's recent data showing that T-cell responses against ENF, they could work some of decoy towards beneficial T-cell targets. And then we have what we call the focused T-cell immunogens, that basically what they try is to divert, to modulate the HIV-specific response. Instead of just expanding what natural HIV already does, is to focus the T-cell response towards what we think could be more beneficial for the host. Some of those focused T-cell responses are based on conservation. That's because of the theory of the Achilles theory, that maybe HIV is able or has some very conserved regions in their HIV genome which are more beneficial for the host. And then there are some other strategies, as the one I'm going to be presenting, the HTI, which is guided not by conservation of the sequence, but guided by immune human data that we have generated over the years. So you have the design here of the HTI. Basically what we did was take large cohorts of HIV-infected individuals with a different range of viral loads without antiretroviral treatment, and then we ask which regions where those individuals with lower viral loads were targeting more frequently than the individuals with higher viral loads. This is what we called by an algorithm as a protective ratio, and then we identified those regions as protective ratio higher than 1. We identified 26 of all of these regions, and then we ensembled them in a continuous sequence. It's a 529 amino acid immunogen. It's more or less covering 16% of the proteome, regions of GAG, POL, BIF, and also NEF, and they are linked by triple alanine. So all these 26 OLPs, what we did was expanded or shortening, considering all the epitopes that are being targeted between those regions, also taking into account CD4 T helper hotspots and the range of HLAs that we wanted to include. And this is our final HTI design. We have expressed the HTI so far in 3 different delivery platforms, all in GMP production already. This is a plasmid DNA that was constructed by the group of Barbara Ferwer at NCI. And then we have 2 viral vectors, MBA and the ChAdOx one, that is the same delivery that was used in some of the COVID vaccines by the Oxford team. What is our pipeline in terms of clinical trials? So the first in-human clinical trial testing those vaccines was the IAS-02 study that was published last year, the results in Nature Medicine. This was a randomized clinical trial, placebo control, and it was testing a very large vaccination regimen of the HDI vaccines alone. That was in early treated population, people living with HIV, and that was a single-site study performed in Barcelona where I work. Then, in collaboration with Gilead, we were able to set up a second study that was the IAS-03. That was also a randomized controlled trial, and here we used a shorter version of the vaccine regimen, CCMM, in combination with a TLR7 agonist. That was Ruxolitinib, that was based on the Verducci's paper, in which when they combined this TLR7 with vaccinations, they show a little bit of delayed viral rebound and a lower set point. That was also a trial performed in individuals with HIV, and that was a multi-site in different Spanish sites. And the trial that is still ongoing is the BCN-03. In that one is also a randomized controlled trial, placebo. Here we are using a T-cell regimen with Chimpadeno MVA boost in combination with a B-cell vaccine, is the SOSIP version 7GP1. 40, with the intention to combine both T-cell and B-cell vaccines in case we can induce some vaccinal effect. This is also a trial performing in people with HIV. This is not early treated. This is individuals that have been treated during their chronic infection. This is performed at our site. All these trials have the same ATI design. It's a 24 weeks ATI. We monitor participants weekly, and we use the same treatment recension criteria, and is as follows: is one determination above 100,000 copies, 8 consecutive determinations above 10,000, 2 consecutive determinations if we show a CD4 cell drop below 350, or clinical symptoms of acute retroviral syndrome grade 3 or higher. As said, both the IAS-02 and 03 are trials that have been completed, and the BCN-03 is ongoing. Last but not least, we have also performed a study, a safety and immunogenicity study in people without HIV that was in collaboration with the Oxford team. It's an open label just to understand if the vaccines were also immunogenic in people without HIV with no pre-existing responses to HDI. So I will go through a little bit IAS-02 and 03 data. This is the IAS-02 has already been published, as I was saying last year. If you remember, there was 2 consecutive regimens with the HDI vaccines. In the first one, we combined DNA prime with MVA boost, and then after a period of rollover, participants were invited for a second vaccination regimen that included now the ChAdOx HDI and last MVA vaccine boost 8 weeks before starting the ATI. And in the IAS-03, we tested a shorter vaccination regimen with 2 ChAdOx and 2 MVAs. And between the first MVA and the ATI, there were 2 rounds of Ruxolitinib administration, which, as you know, is an oral drug with dose that at 6 milligrams. As said before, these both trials were performed in early treated population. In total, 88 participants were included, randomized 2-to-1. So study populations were really comparable between both studies. Basically, men were included, MSM population, time from estimated HIV acquisition to treatment initiation was around 2 months, 16 days in median. And you see here the distribution of the FIVIC stage at treatment initiation. You see that it's mostly early treated population rather than acute. It's mostly FIVIC 4 and 5. Okay, which I think that for the therapeutic vaccination regimens is kind of favorable because they have already mounted a T-cell response during primary infection. It's not that it's a FIVIC 1 stage without no T-cells around. And you see that they have pre-treatment viral loads very high and good CD4 cell counts at study initiation because they were early treated. In terms of immunogenicity, we try to set up exactly the same immune assays between both trials to make them, to be able to compare them. And this is the results of the ELISpot interferon gamma assays in which we tested HDI peptide pools and also peptide pools covering the non-HDI regions. As I was mentioning, HDI covers around 60% of the viral genome. And as this is people, all the participants are people with HIV, we know that they already have other HIV T-cell responses, and we wanted to address the role on the potential outcomes. So in all the ELISpot data, it's included both HDI and non-HDI. You will see over all the slides placebo recipients depicted as blue, and the vaccine or vaccine plus besatolimod recipients depicted as red. And you can see here the comparison of the immunogenicity kinetics. In ILX02, we saw a first great boost after the first MVA shot, okay, that rapidly waned, and it was consequently increased over the consequent vaccinations. And in the ILX03, after the first boost of MVA, and then with besatolimod, we saw a progressive increase of T-cell responses. The good thing is that at the moment of ATI, both trials had equally non-significant differences, responses towards HDI. You see in the below pies the distribution of what we call the immunodominance towards HDI. So in the red pie, the red part of the pie, you will see out of the total HIV-specific T-cell responses, how much of those are targeting HDI. If you remember, 60% of the viral genome, and we are able to switch all these immunodominance patterns to 50, 60, 80% of all the HIV-specific responses being specific towards HDI. When we go into the breadth, that's also very important. We are talking about a synthetic immunogen. We want to make sure that all the things that we have included in the immunogen are being expressed and are being immunogenic. This is what we show here. This is the breadth, the cumulative breadth. That means the different targets of the HDI that are being triggered during the immunization. And you can see that all the GAG regions, all the POL regions, and even the BIF-NEF, which is a very small part of our immunogen, is being targeted over the vaccination regimen in the actives. And here is the decays on the HIV reservoir. Of course, we are not including any latency reversing agent. Also, besatolimod is not expected to induce viral reactivation, but it was not surprising to see that the decay curves were exactly the same between the placebo and the actives. You can see that in the O2, the decay of the intact HIV reservoir was a little bit stronger than in ILX03, and that's just because in ILX02 the participants were starting in the study after 1 year of treatment initiation over 3 years, so that's the natural decay over treatment initiation, while in IL03 they had been suppressed for longer. These are the ATI rebound kinetics, and as it was mentioned before, you can see that more or less the kinetics are very similar in terms of when the virus is rebounding. How is it starting the exponential increase? Then it reaches a peak between 3-4 weeks after viral rebound, and then it goes to the set point. And what it's clear is that we tend to see more actives compared to the placebos remaining off treatment for longer periods of time. But it's true that when we put this together in the survival and we pull the data from both trials, well, you see there's a little bit of signal between the actives versus the placebo, which is a little bit stronger when we remove the noise of the participants with beneficial HLAs, but well, it's not that high. However, we would like to see which are the correlates of that potentially efficacy signal, and then we put in an individual level first all the demographics, all the viral reservoir parameters that we have, and all the HDI immune parameters. This is just a wrap-up of some of those. We have measured many others, okay, but basically the significant ones is if you have lower plasma viral loads before treatment initiation, during your acute infection, probably you're going to behave better if you receive the vaccine, not if you receive the placebo. Lower viral reservoirs also can have an impact, but you can see that there's a strong efficacy signal or correlation signal with the magnitude of HDI-specific responses at the moment of ATI interruption. When we put all this together in a multivariate analysis, even if when we fix the effect of the plasma viral load and the reservoir, you see that there's a 12% of increase for each 100 SFCs that you have at ATI start in terms of magnitude HDI. So the magnitude HDI is the covariate that has a significant impact on the outcome. When we try to understand how probable is or what are your odds of remaining off treatment for more than 12 weeks. Okay, So if that's magnitude, can we define a threshold that can help us to understand if you're going to control or not? So when we put this on a ROC curve, which means that we are trying to address how well is that predictor in the outcome. I don't know if you're familiar with this type of curves, but basically if your line is during the diagonal, that means that your marker is totally random effect, and the closer to the edge that your value is the better that it predicts. So an area under the curve of your ROC closer to 1, that means that it's the best predictor. And we found out that having a magnitude of HDI-specific responses around 835 or higher was able to predict with a very high sensitivity and with a very high specificity your odds to remaining off treatment. Having a predictive value of 73 means that if you make it to that threshold, 73 of participants will be out of the ATI for longer than 12 weeks without treatment resumption. So when we put this in the survivals, now we see the signal, and I hope you believe that there's a signal, both in having a delayed viral rebound. You see that individuals on red, meaning individuals, vaccinees that made it beyond 835 at the moment of ATI with a simple ELISpot assay, just measuring HDI-specific responses, 50% of those individuals had a delayed viral rebound in terms of how many days they had their first detectable viral load. They had a slower rebound, meaning that almost 50% of individuals had a delayed time on reaching a viral load of 10,000, and they were 70% of individuals with higher than this threshold being off treatment for longer than 12 weeks. Out of those, 25% were with viral loads below 2,000. So I hope you'll be with me concluding that HDI vaccination alone or in combination with vesatolimod induced both equally strong and broad HDI responses, but we didn't have an impact on viral reservoir. The magnitude of HDI appears to be a good or potential correlate of an improved viral control when it comes to ATI, and of course we need to further validate the predictive value of these thresholds in upcoming future trials. We need to elucidate factors that contribute to a better vaccine responsive among all our vaccinees. Why 50% of our vaccinees made it to the threshold, and why 50% of our vaccinees did not make it to the threshold, what it matters there, and how we can improve that effect. We need to reproduce the results in much more diverse population. I think is our missing strategy. We are failing always in the early treated cohorts to include women, and that's really an issue. So we need to move to other populations because in our setting it's going to be very, very complicated. And I hope we are convinced that all the new strategies, combination trials in the cure field could use HDI as a potential good T-cell backbone to enhance the vaccine immunogenicity and to increase that efficacy to a critical clinical endpoint. And with that, I would like to thank all the people that has been involved in my clinical team at IRC Caixa in developing in the first years the vaccine, and now with IELIX giving all the support for all these clinical trials, all the people from Gilead has been supporting our program, and all the investigators from the IELIX 03 studies. So thank you very much.
Okay, thank you, Beatriz. Due to time constraints, we will have all QAs at the end of the session. And now I will introduce our next speaker. She is Julie Ake from the US Military HIV Research Program, and she will be talking about immunotherapies in acute cohort.
Thanks so much to the organizers for inviting me to talk on this topic, because we focus a lot on acute cohorts at the Military HIV Research Program. And our mission includes the development, we're probably best known as an HIV vaccine organization. But in the attempt to really understand what are the earliest events in HIV infection that have to be blocked by a vaccine, many years ago we started to do work in acute infection, which has then been an opportunity to leverage for the remission agenda. And we're lucky to be doing this in the context of where we work overseas, also administering the President's Emergency Plan for AIDS Relief. So we're able to engage with communities and provide service delivery where participants accept risk for being part of our trials. And so a lot of our international footprint includes collaborations in sub-Saharan Africa and long work, of course, in Thailand, now expanding to the Philippines and Southeast Asia. And these are the places where we've really focused our work in acute HIV. With two primary cohorts. The first is RV217. This is led by Merlin Robb. This study is over now, but what this study did was it actually followed people twice a week who were at very high risk for acquiring HIV infection. And because of that, they were able to capture the earliest events in HIV infection, and this was published in the New England Journal years ago. I would just point you to the shape of that curve, right? We'll see it later. It was really important to be able to characterize this in the global settings of East Africa and Thailand, where this study enrolled. 254 is ongoing in Bangkok, Thailand. This study enrolls and identifies participants in early HIV infection through a different mechanism, essentially screening people who come for HIV testing for acute infection, and then after those individuals are identified, consenting them oftentimes not just to early ART, but for the provision of additional specimens through the optional procedures that you see here, and then the initiation of very early ART, with large numbers of individuals enrolled and starting therapy in very early HIV infection, including stages FEBIG 1 and 2. And so if you actually look at what is the impact of that, this is an old slide, but it showed the early parts of 217 were before test and treat started. And you can see, even just in total HIV DNA, the big difference, about 300 times, right, that early treatment of HIV makes in comparison between the two studies. So one would imagine that if you have a cohort that treats people very early, they have a low reservoir, these are a good population to perform cure interventions and analytic treatment interruptions. And indeed, we've done that. We've done that 4 times. In the studies that you see here, whether it's just interrupting individuals who were treated very early in FEBIG 1, combination with vorinostat, hydroxychloroquine, and Maravirac, which was 409. And then 2 immunotherapy interventions, one a therapeutic vaccine with Janssen, and the other one VRC01, both in treatment interruption. And you'll see here that the only situation where we found a delay in time to rebound were those 2 immunotherapy interventions, the monoclonal, that was presented at this meeting initially by Trevor Kroll, who's here today, and in RV405, which was the therapeutic vaccine. But what we did learn is that we didn't cure anyone, right? We just delayed time to rebound. And individually, these various interventions do not induce remission on their own. So today we're really focusing on combinations, as the rest of the field is, either in ATI with interventions to boost immune responses, like RV582, I'm not going to talk about that today, but also by intervening earlier during acute infection. And that is what I'm going to focus on today, 2 studies, RV398 and 550. So I won't go through this very much. We've heard a lot about the rationale for intervening during acute HIV infection and early HIV infection, starting with some really important non-human primate data coming out of Mal Martin's lab, with some viremic control in non-human primates found to be CD8 T-cell mediated, and sort of the first evidence of the vaccinal effect many people are discussing here, at least as related to the early intervention with MABs in early HIV infection. Then we've seen a lot of the great trials coming out of the Rockefeller program, which I would highlight here, included demonstration of sustained virologic control in ATI of people living with HIV early, who are treated in early infection. Okay, very relevant to the work that we do. And then, of course, the recent Nature Medicine eClear publication, which showed that starting antiretroviral therapy along with 3-BNC117 plus or minus romidepsin in people who had underlying virus that was sensitive to 3-BNC117, those individuals were more likely to maintain ART-free virologic control in an ATI as a subset of individuals underwent in that study. So as those studies have been going along, we've been completing RV398, where we gave VRC01 during acute HIV infection. I'm not going to talk too much about VRC01. People understand this molecule that's been very well studied in non-human primates and people in the treatment and prevention setting, notably being demonstrated to prevent HIV in people who have underlying sensitive virus in the AMP trials. In this study, we actually treated during acute HIV infection. So identified individuals first in 217, and then as that study ended, moving to 254. And we enrolled people with the criteria that you see here during that acute HIV infection and randomized them to one of three groups, just 8 each, pretty small study. This was done in 3 countries in East Africa and in Thailand. So group 1 received placebo followed by, along with immediate ART. And group 2 received VRC01, 40 mg per kg IV, along with immediate ART. And group 3 received the VRC01, and the ART was started 7 days later. So you could look at the independent impact of the MAB. And then the objectives were to look at safety and then the impact on viremia in the first 7 days. The baseline characteristics I would just like to highlight, because we work in high-risk populations in East Africa, we're able to enroll women. So we had almost 30% of cis women enrolled here, and about half of the people who were enrolled in phoebic stages 1 and 2. I would note that generally the demographic characteristics were well distributed among groups, and that people were enrolled with about 5 logs of virus. A lot of work goes into one safety slide, but it was important to note that the VRC01 was well tolerated across the treatment arms without deaths or SAEs. We did have an SAE in someone who received both the MAB and subsequent ART that was associated with a post-LP headache. And then subsequently, we did have one quickly self-resolving severe transaminitis in the MAB plus subsequent ART group. Okay, this is generally quite well tolerated. So what happened with the viral load? So what I'm showing you here are the virologic curves, and the group 1 is in the orange with the placebo and immediate ART, group 2 in the green, VRC01 and immediate ART, and the blue had the delayed ART. And you can see here that there was a significant reduction in mean viral load from baseline to day 7 with immediate ART groups. And so both immediate ART groups compared to the delayed ART groups had faster viral decline in the first 7 days. The median time to virologic suppression was the shortest in the combination group of MAB plus immediate ART, and then the placebo plus immediate ART, the second shortest, followed by MAB plus the subsequent ART, and that was not a statistically significant trend, but you saw this also with the area under the curve being the lowest in the combination group as well. This is just a blow up where you can see that the blue group with the delayed ART did have the higher virologic curve, but it's blunted, if you remember what we saw with 217. This is just the statistics showing that the only statistical differences were between the ART-containing arms and the delayed arm. So what about the viruses that we enrolled? Well, they reflected exactly where we enrolled, right? So, I mean, the viruses of the participants we enrolled just reflected the countries where they're from. So in Thailand, we had B and CR4-1A, and then in East Africa, we had A and C. Most of the infections were single founder infections, but 4 were multiple founders, and 1 infection was actually with 2 independent viruses. The neutralization sensitivity was important. We found that less than a third of the viruses were highly sensitive to VRC01. So if you take a look at also the distribution, unfortunately, many of the most sensitive viruses, I'm showing here IC80s, and the lower the bar, the more sensitive the virus. The most sensitive viruses were disproportionately in the placebo group. So that sort of limited our ability to look at the viral neutralization sensitivity as it impacted the reservoir viremia and other measures. We did not see a significant difference in cell-associated total nucleic acid, and this is RNA plus DNA, and then some very fresh data coming out of our low copy number viral load assay essentially did show us that in the immediate ART arms, we had more episodes of the target not being detected at the durability time point in the ART-containing arms compared to the other. We did not see significant differences across groups. In total DNA, perhaps a trend, with the combination group being a little bit lower, if you see there at day 168, but this was not seen in the integrated DNA, and it was not clear that there was an impact of neutralization sensitivity on this, and that was, of course, limited by the distribution across groups. So I would focus you here on the GAG-specific T-cell responses for the CD8s in the interferon gamma and the combined interferon gamma TNF alpha groups, where you can see at the durability time point in the mAb receiving individuals, there did seem to be average increased responses in those receiving the product compared to placebo. I think this is very subtle, but consistent with some of the other findings that we've seen. And then, as we know, VRC01 has FC effector function, and we saw that immediately after infusion with ADCP and ADNKA being obviously higher in people who got the mAb. Interestingly, we wanted to look at activation and inflammation, and Diane Bolton's group took a look at peripheral blood immune cell phenotyping, and they found really what you might expect in terms of activation in T lymphocytes. You can see here that CD4s were actually pretty stable in terms of their activation, and that CD8 T-cell lymphocytes, their activation was largely correlated with viremia. But for monocyte and dendritic cells, we saw a different story, which was pretty interesting. There was strong monocyte activation in acute infection, and it rapidly decreased following ART, fully resolving by day 28. But interestingly, there was still a substantial reduction of monocyte activation by day 7 in the group that had VRC01 monotherapy. Whereas for T lymphocytes, there was not decreased activation until there was ART started. But here, for monocytes, and we also saw this with dendritic cells, which I'm not showing this data, we found that there was decreased activation in the context of mAb alone. And then similarly, Rashmi Thomas looked at bulk transcriptomics, and she found that starting at day 3, and then shown here at day 7, so this is the time period where that one group got VRC01 alone, they found an inflammatory signal of upregulated interferon stimulated genes. We found that diminished in this delayed ART despite ongoing elevated viremia. Moving on to a mucosal compartment substudy, we were happy to have volunteers who participated in these optional collections, and thankfully it ended up being 5 volunteers in each group. We were able to detect VRC01 in everyone who got the infusion, and we didn't see an apparent impact of the monoclonal on CD4 T cell populations, but we did see that post-infusion, the VRC01 levels directly correlated with the frequency of rectal CD4 CCR5 positive cells. And so those who got the placebo, you can see here very small numbers, they had a significant drop that was actually maintained in people who got VRC01. And furthermore, VRC01 levels were inversely correlated with frequency of cell cycling and activation, as you see here by Ki-67. So again, we're seeing some decreased activation in the mucosal compartment. So with this study, we generally were able to show that VRC01 was safe and well tolerated. The viral load was decreased significantly in the immediate ART groups, and generally the combination group did the best as you measured all of the various outcomes of viremia. We were challenged by decreased sensitivity of circulating viruses to VRC01, which is not a surprise to anyone anymore. We see a trend towards increased CD4 T cell GAG responses. FC effector functions are known to be characteristic of VRC01, which we saw in our study, and we were very increased in the decreased inflammation and activation in mAb recipients. So please stay tuned. We are going to be doing transcriptomic analysis of sorted cell populations to elucidate that further. So what's next for us? This is just an ongoing study. We're very interested in the superagonist IL-5 complex N803 with Immunity Bio. This has demonstrated important reservoir effects in non-human primates and also resulted in viral control in a subset of animals when it was co-administered with monoclonal antibodies. The clinical experience, there is a known local reactogenicity with this molecule, and this was observed in its initial phase 1 HIV trial, but was also associated with viral reactivation and reduction in the latent reservoir. So this was in treated chronics. We were interested to administer it in acute HIV infection. So this is an ongoing study led by Dr. Kiat in Bangkok with the hypothesis that initiating N803 during AHI will be safe and lead to a reduction of reservoir in the lymph nodes. And so we recruited individuals willing to undergo lymph node biopsy in the RV254 cohort. There are 3 left to enroll. We're seeing the expected local reactogenicity. Everyone's warned about it in their consent forms. They're all coming back. It's being quite well tolerated, even though these are some, pretty significant, typically injection site redness events. So we'll look forward to seeing the outcomes from that trial. And of course, our next efforts, presuming that continues to be positive, will be to combine some of these interventions. I would share that these acute and early treated cohorts are really promising for remission trials. They may facilitate a precision medicine approach if you have a very limited reservoir with a limited diversity of that reservoir for individuals who are treated so early that may allow for a precision cure approach. And then when you're conducting these ongoing cohorts, they allow for the early delivery of immunotherapies. And I think this is something that is very doable in the context of low and middle income settings. We certainly did this in 398. So be open for the possibilities of what's feasible there. And while we definitely need better global reservoir assays, they're coming. We need to be doing these trials now. And that's all I've got.
Thank you very much. So the next speaker will be a flash talk on immunotherapies in non-human primate by Ofam Okoye from Oregon National Primate Research Center, USA.
All right, I want to thank the organizers for the invitation. It's great to be here in Brisbane. And these are just my disclosures. Okay, so just very quickly go through some of the things we've been trying to do in non-human primates to try and understand how we can use immunotherapy to facilitate post-hoc control. And just to highlight, as we've heard for most of the morning, that in most cases when individuals stop ART, virus rebounds, and rebound often doesn't lead to control, and only in few cases you have rare individuals that can control viremia by themselves after ART interruption. And for us, we're really interested in trying to understand how T cells intercept this post-ART rebound. And I guess a major challenge for CAT cells is the idea that when infection is rebounding, you have multiple foci of infection that is reactivating in multiple different sites. And so unlike during primary infection, where you have initial founder viruses that are probably in a very few areas of intercept for the CAT cells, for post-hoc control, when you have a systemically distributed virus that is in multiple different tissues, you're really challenging CD8 T cells to be able to intercept in these different sites and in these different tissues. And so one of the things we did try, that we published a couple of years ago, is because we wanted to understand what was really the limitation of CD8 T cells at the time of that initial viral rebound after ART was stopped. And so to try and answer that question, we had non-human primates that were infected with SIV MAC239, which is a barcode, 239M, which is a barcode virus developed by Run and Kill. And what we did was that we asked if we take away classical MHC class 1 restricted CD8 T cells and stop ART, can we actually identify what differences we see in terms of post-viral rebound. And that gives us an idea of what the limitations of CD8 T cells are. Then we can begin to think about interventions to challenge that. And one of the major things we saw was that when we take CD8 T cells out and we look at these early viral dynamics, what was the frequency and the magnitude of rebound in animals that had no CD8 T cells versus animals that didn't, and we didn't actually see any difference. We actually saw that, at least the time to rebound was very similar. The initial early kinetics of rebound was very similar. But when we did have viral amplification and spread, when there is post-op viremia, we then begin to see an effect of not having CD8 T cells around. So in other words, the T cells are there and they're potent. They can have an impact. Animals that don't have CD8 T cells, you can see, are in red. So viral load set points were almost a log or two higher than animals that did have CD8 T cells present. So it really suggests that there's really this dynamic mismatch between this initial virus amplification and spread and a lagging immune response. So the immune responses kick in later, but they're not fast enough or of the same magnitude or effective function at the time of rebound to be able to intercept that rebounding infection. We then went on, I think, and this was, we reported this last year, to ask what would happen if we augmented CD8 T cells during this rebound period, with the idea that perhaps, some of these cells might be exhausted. So if we use an immunotherapy to try and help reverse exhaustion, can this facilitate an earlier intercept of these rebounding infections? And so what we did, we designed an experiment where we gave PD-1 blockade, starting in some animals just before art release and some animals at the time of art release. And we asked the question, do you see an earlier intercept of rebounding infections if indeed immune exhaustion was really the major focus driving the slower or lagging response? And for the most part, what we saw, we didn't see any difference in that initial time to rebound. So whether PD-1 was on board or not, we saw that all animals rebounded around the same time, and that initial early viral kinetics was very similar between all groups. However, in animals that did receive immune checkpoint blockade, we then began to see that there was an evidence of early post-hoc viral control. So then again, when the virus comes out and there's amplification and spread and there's antigen-driven activation and expansion of these CD8 T cells, if you have PD-1 on board, then it actually facilitates more of an effective intercept of the response. So really, the question really we've been trying to answer is what happens if we mobilize CD8 T cells earlier during that recrudescent infection? Is that going to be able to allow us to then be to intercept the virus earlier? And if we intercept the virus earlier, then would we be able to facilitate post-hoc control? And as you can tell with most of the post-therapeutic vaccine studies we've been hearing about, most vaccine studies enhance the immune response, enhance the breadth of the immune response, but that early intercept of rebounding infections at the time of art release has really been lacking. And so really the question is, well, what happens if we play around during that intercept time? So in most cases, I think what we're seeing is that the virus just really takes over, and the virus always seems to get ahead of that rebounding infection. And so can we shift that balance? Can we actually make the immune response to be more potent and more active at the time of intercept, and would that be sufficient for control? But there is a challenge with this, and one of the things we've seen recently is that we've recently asked a question with IL-15. This is NA20 is a fusion protein that has both IL-15 and CD20, and we're trying to ask what happens if we disrupt follicles at the time of art release. And just to highlight that when we apply IL-15, either during full art suppression, you get really good expansion of memory T cells, which are target cells for the virus. If you give IL-15 during full art suppression, we don't see any increased activity of viremia, but when we give it at the time of art release, we actually see an expansion of memory T cells, and we also then begin to see an increase in post-art viremia. So it really does say that there is a challenge with intervening at this important time, because while most of these immunotherapy interventions are trying to enhance CD8 T cell responses, but they also have an impact on CD4 T cells. So the question we wanted to ask, which I'm going to discuss very quickly, was whether if you use neutralizing antibodies at the time of rebound, could this really help us slow down the pace of reactivation of the virus? So if you slow the rate the virus is reactivating from latency, this gives you an opportunity to allow to see your immunotherapy interventions to get ahead of that rebounding viremia, with the idea being that neutralizing antibodies have a slower decay of washout relative to ART, which decays at a much more faster rate. And recently we've been able to get 2 neutralizing antibodies that are effective against SIV MAC239, which is typically a very neutralization-resistant virus. One of them was identified by Dennis Burden's group called K11, and the other was Mario Roda's group who identified ITS 103.01. And in a study we did, in a pilot study, we gave these 2 neutralizing antibodies in combination at the time of our release, and we showed that there was a significant delay in time to rebound. So the question we wanted to ask is, what happens if we combined these neutralizing antibodies at the time of our release in combination with immune checkpoint blockade? Because in the previous study we showed that we could enhance early post-ART control, but now what happens if we slow down the pace of rebound and apply a PD-1 blockade? Can we facilitate, will this be better to get better control? And so again, we gave animals, we infected animals with SIV MAC239, put them on ART early in acute infection, we suppressed them for a long time. We started anti-PD-1 just before ART release, and then at the time, 3 days after ART release, which is the time when ART is washed out, we then gave the combination of K11LS and ITS 103 to one group, and another group only got the combination control antibody by itself, and then we had a control group. So what happened? Well, again, what we saw was that when we combined, when we gave animals that received the neutralizing antibody, whether they got it with PD-1 in combination or by itself, we actually saw, again, a significant delay in time to rebound, as we would expect. However, when we looked at the antibody combination group that got a neutralizing antibody with PD-1 relative to neutralizing antibodies by itself, we didn't actually see any difference in time to rebound. So it's telling us that whether you had the neutralizing antibodies on board or not, that there was no difference in what PD-1 with PD-1. These animals were all infected with the barcoded virus, so we can actually look at how many barcodes were contributing to rebound to give us an idea of the clonal complexity of the rebounding viremia. And we didn't actually see a difference when you had PD-1 on board or with antibodies, or whether you had antibodies by itself. And this is just highlighting how with deep sequencing, we can actually then look at different clones that are contributing to rebound, and we didn't see any difference between the groups. And for the most part, what we saw was that time to rebound really was dictated by the decay in antibody titers. So once the antibody titers dropped below a certain threshold, that's when we began to see rebound viremia. And in some animals, we saw that antibodies decayed almost to undetectable levels before rebound occurred. And in some animals, actually, we saw that the titers were not completely decayed before we saw rebound. So for the most part, in time to rebound was really dictated not by PD-1, anti-PD-1 treatment, but decayed antibody titers. And overall, when we compared animals that got PD-1 with BNAbs versus BNAbs by themselves, we actually saw that with anti-PD-1 on board, we saw quite a few animals that had improved post-ART viral control relative to when you had BNAbs by themselves. Normally, we typically try and exclude animals that have protective MHC alleles, but these studies were done during COVID, and there was really limited time to select animals. So we had some animals that had protective MHC alleles, and they were evenly distributed between the groups. And what we see actually is that when animals have a protective MHC allele, in other words, they are predisposed to develop optimized CD8 T cell responses. When you have PD-1 on board, we actually saw that these animals actually had even better post-ART control. So it really says that perhaps that PD-1 actually can augment CD8 T cell responses to really improve control, especially if those responses are really predisposed to control viremia really well. And this was even more effective than what we saw in the other groups. So just to summarize, augmenting CD8 T cells at the time of with PD-1 blockade at the time of our release really didn't have any impact on how the virus rebounds. And I think one of the major points I wanted to stress is the fact that what we saw was that for the most part, time to rebound was dictated by decaying antibody titers. And it tells us really that what we are seeing in our model, at least, is that neutralizing antibodies, once they decay below a certain threshold, we see rebound occurring. And this is it's telling us antibodies are really behaving like ART. It's limiting viral spread. And once they decay, we see rebound kinetics occurring as we would without it. And just to say thank you to a whole bunch of people. Most of these studies are funded through the DARE Collaboratory, and this is the DARE study team and leadership, and everybody in my lab, and thank you.
So our next speaker is Siwin Chen from the University of Hong Kong, and we'll be talking about vaccination against PD-1.
I want to thank the meeting organizers for giving me this opportunity to share our results on PD-1-based DNA vaccine for HIV functional cure. About 10 years ago, we reported a strategy by using soluble PD-1 infusion with HIV antigen to target dendritic cell. So this idea actually was pioneered by Ruxolitinib by using the anti-DAC205 antibody to target HIV vaccine antigens to the dendritic cell. But in the parallel comparison, we found the soluble PD-1-based targeting can specifically, actually more uniquely engage the intracellular compartment for antigen cross-presentation for inducing a high frequency of CD8 T cells. So this is a difference compared with the anti-DAC205-based targeting. Then, in order to test the potency of the CD8 T cell induced, we used the cancer model in immune-competent mice. And this is in a therapeutic setting when the animals pre-implanted with a very malignant mesothelioma. And after using the PD-1 targeted vaccine for therapeutic use, we were able to cure all the animals in this study, suggesting the quality of the CD8 T cells are much different compared to traditional vaccination. So then we did two sets of the study using the non-human primate models. In this study, basically, we using different regimen to immunize the animals, then wait for over about 30 weeks to mainly focusing on the memory response in control of the subsequent viral challenge. And both studies consistently show that the vaccine-induced CD8 T cell be able to control the set point viremia very significantly and also achieve the long-term control of the viral replication in all the vaccinated animals. So then the vaccine induced the CD8 T cell by testing using multiple assays as basically broadly reactive polyfunctional CD8 T cells, especially the effector memory CD8 T cells, targeting multiple epitopes by secreting multiple cytokines, and that's the quality of the CD8 T cells elicited by this vaccine approach. And most importantly, after the viral challenge, we can see the viral challenge can recall the memory response, mainly the CD8 T cell high frequency up to like over 10% of the CD8 T cell. In this case, is about 7%. So this is a major difference compared to other traditional vaccination. So then we did the CD8 depletion, want to see whether it's truly the CD8 T cell control the viremia, and definitely all animals rebounded, suggesting that. And after 6 years after COVID-19, we paid the visit to the group, the first group of the animals, and 2 of them still survived for over 6 years. And recently we did also the functionality analysis to evaluate the CD8 T cells. They still persistent over 6 years of the time. And you can see the survival advantage is very, very important. And after this non-human primate work, we're now generating a human PD-1-based vaccine by using the mosaic antigens based on over 500 GAG sequences derived from Chinese patients. And then we did the primate study to evaluate the immunogenicity, and similar broadly reactive CD8 T cells were elicited in monkeys. So after this information obtained, we also use the peptide derived from mosaic antigen among currently people living with HIV, suggesting basically the results, suggesting the cross-reactivity do exist among the individuals living with HIV. So that's why after 10 years of efforts, recently we finally launched the human trials. And so far, this is a double-blinded, placebo-controlled, and also dose escalation design. So far, we already finished the low-dose vaccination in this group, and all the safety evaluation satisfied, and we got the approval. Now, currently, we are doing the mid-dose vaccination. And hopefully, in future, I can share the results. And we are also planning to decide at what time point we can stop the antiviral therapy, because current requirement is all the patients, even receive the vaccination, still have to under the antiretroviral therapy. So that's what I want to share. And at the end, I want to thank all the collaborators and also my team at the University of Hong Kong and also the funding agency. Thank you for your attention.
Thank you very much. So now presentation from Diane Schonder from Peter Doherty Institute here in Australia on favorable transcriptonic signature of HIV-specific CD8 T cell in response to a single anti-PD-1 dose in people living with cancer and HIV.
Thank you very much. And thanks for the invitation to speak here. Okay, so this project is led by Celine Gupsa in Sharon Lewin's laboratory at the Peter Doherty. I'm also from the Peter Doherty in Melbourne. My name is Jan Schröder, and I'm a computational scientist who did the bioinformatics analysis on this. So let's first talk about the role of PD-1 in HIV and cancer. So PD-1 is expressed on exhausted T cells and impairs T cell function in cancer, but also in HIV, as we learned. Latent HIV is enriched in PD-1 CD4 T cells in blood and lymph nodes from people on ART, and anti-PD-1 has been shown to reverse this HIV latency. Anti-PD-1 can enhance HIV-specific CD8 T cell function in people with HIV on ART, and this is what we're interested in this study. So it could have an impact in the role of treating HIV, but the impact of anti-PD-1 on HIV-specific T cells is so far poorly understood. So what are the predictors for a successful response to anti-PD-1? So first, there's been studies in mice that have shown that a subset of CD8 T cells, the precursor exhausted TPEX cells, give rise to exhausted T cells, or TECs, which are essential for the long-term maintenance of the immune response. And second, in humans, there's been studies in cancer which have found individuals who respond to anti-PD-1 treatment have an increased frequency of these TEC cells and improved clinical outcome. So therefore, we have hypothesized that anti-PD-1 will reinvigorate HIV-specific T cell exhaustion in people living with HIV on ART and lead to a decline in the HIV reservoir. So we aim to characterize changes in CD8 T cell subsets and the transcriptome of HIV-specific CD8 T cells and the HIV reservoir following anti-PD-1 treatment in people living with HIV on ART and cancer. So our data comes from this study, the AMC 95. And let me double check that I get this right. So the AMC 95 was a USA-based prospective study of anti-PD-1 and anti-PD-1 plus anti-CTLA4 immune checkpoint blockade treatments in people with HIV on ART with a range of malignancies. And for this project, we analyzed samples from 8 participants who only received the anti-PD-1 treatment. Treatment cycles were 2 weeks long, and sample collection was performed on the day before anti-PD-1, the day after, and on day 8. We have the blood samples of these participants from 8 donors from various time points. And for simplicity, we're going to summarize these time points as baseline, single dose, and multiple doses. So what we did is look at CD8 T cells in these participants, in the blood cells. We used various HIV-specific tetramers to pull out HIV-specific T cells. that you can see here. And we found that in 4 of the 8 participants that there was an increase in these tetramer positive cells at the first dose time point. So we set out to do single-cell sequencing on these CD8 T cells. And here you can see the entire data set coming out from this project. This is a UMAP which shows single cells from all the individuals and all the time points across this project, and their cell identities on a UMAP cells cluster together in a similar area if their transcriptomes are similar. And here you can see our annotated cell identities. This heat map shows a fraction of this data where you can see some of the genes expressed in these cells on the y-axis and single cells on the x-axis, and you can see the differences in these cell types and why we identified them as such. Then we looked at the composition of these cell identities across time and across participants, and we found a striking increase of TACs, the exhausted T cells, at the first dose after the first treatment. And we looked further into this by participant and found that this increase in TACs frequency was significant in 3 out of the 8 participants, going together with a decrease in TTM cells, whereas no such effect was seen in the remaining participants. So we therefore labeled these 3 as the responders to the treatment, and the others non-responder, shown in green and purple from here on. We then also looked at the transcriptomic differences of single cells at the first treatment time point and baseline, and we found a strong signature here in the TCM compartment. And interestingly, the same 3 participants that we had seen here as standing out in increasing in TACs were driving this transcriptomic differences in TCM, which was related to interferon-stimulated genes, including STAT1, MX1, IF7, and IF9. And looking at the CAC pathways and GO terms, we found interesting signatures here relating to interferon signaling, immune response, and HIV-1 viral life cycle. Okay, and then we looked at the levels of HIV RNA and DNA, but we did not see a significant change here over time or between responders and non-responders in these numbers. But of course, our study design is pretty small, so it has to be interpreted with caution. Okay, so in summary, following a single dose of anti-PD-1 in people with HIV or not, we saw a couple of things. We saw this expansion of TACs and decrease in TTM in some of the participants. And those same participants had a distinct transcriptomic change in the TCM transcriptome in response to HIV-1 viral life cycle, interferon stimulation, and antigen recognition. We did not see a change in the reservoir of viral RNA or DNA in correlation with this. So our findings provide novel insights into the impact of anti-PD-1 on antigen-specific T cells, which were previously not possible to study in cancer alone. Here's a summary for the community. So why did we do this? We wanted to know whether this anti-cancer drug can have an impact in the context of HIV as well as pathway to a cure. So we collected blood from people living with HIV on ART who also had cancer and were part of the clinical trial to receive anti-PD-1 treatment. We sorted the blood into the cells that recognize HIV and analyzed them on a single cell level doing RNA sequencing. We found that in 3 out of the 8 participants there was an increase in cells that indicated a response to the treatment and a reinvigoration of the immune system fighting HIV. Why is it important? Because it allows understanding how anti-PD-1 can reinvigorate the immune system and why only some people respond. And this will allow us to better tailor the use of this treatment as part of HIV cure strategy. So let me acknowledge there's a lot of people involved in this, so let me just highlight Sharon Lewin and Celine Gupta, who have been driving this project with a lot of passion and energy, and it's been a pleasure to work together. And of course the participants in the AMC 95 study, without whom this wouldn't be possible. Thank you very much.
So the last speaker of this session is Monica Rees from the Emory University, and she will speak about the role of Ruxolitinib on the HIV reservoir.
Thank you so much for the introduction, and I'm so glad that you could all be here with us today. I would like to jump right into the reservoir decay in the A5336 human trial. So as we can all recognize, the HIV-1 reservoir is probably the largest barrier to cure efforts, and our group has previously characterized this profile of ongoing inflammation in people living with HIV that contributes to reservoir maintenance and immune dysfunction. And the goal of our group is actually to repurpose FDA-approved immunomodulators, specifically JAK1/2 inhibitors, to reset this immunologic milo that is the ongoing inflammation that is a key driver of this is the JAK-STAT pathway. And so, speaking of the JAK-STAT pathway, I just want to give you a nice little summary of how this works. So you have viral recognition of the host receptor, which generates pro-inflammatory cytokines, and this activates the JAK-STAT pathway, which these Janus kinases will then phosphorylate STATs that dimerize. They will undergo nuclear translocation and bind to various promoter regions for pro-inflammatory genes, creating more pro-inflammatory cytokines. And as you can imagine, this establishes a positive feedback loop, which really contributes to that reservoir maintenance and immune exhaustion. So in the study, I'm going to speak about Ruxolitinib. The intervention here is a JAK1/2 selective inhibitor, and it stops this pathway in the beginning. And so it is FDA approved for various other indications. It is orally bioavailable with twice-a-day dosing. There are interactions with things like CYP3A4 inhibitors, but it is approved for chronic long-term use, including in children greater than 12 years of age for some indications. It is hepatically cleared and BBB penetrant. So just to give you the setup of this trial, it is a phase 2A multi-site randomized trial with 60 participants between the ages of 18 and 75. They were on an ART regimen for at least 2 years and continually virologically suppressed with no other significant medical conditions besides HIV or hypertension. Our group has previously studied this trial, looking at the primary and secondary endpoints of safety and in activation of the immune system. And so what I'm going to talk to you today specifically is looking at the reservoir decay. And so I mentioned these patients, 60 of them were randomized into the two groups. 20 went into the control group, where for the entire 12 weeks of the study, they received only their regular ART regimen, whereas the other 40 went into the treatment group. And from week 0 to 5, they received the intervention Ruxolitinib at 10 milligrams orally twice a day on top of their regular ART regimen. At week 5, that intervention was removed, and they were followed out to week 12, still on their regular ART regimen. Peripheral blood samples were taken at week 0, 5, and 12 in this study. So the first thing I want to talk about is the reservoir. So we measured the integrated proviral DNA using IPDA assay. And here I'm showing you what that looks like over the time of the study. And as you can see, there is no significant decay in the control or the total Ruxolitinib treated group. So what we thought was, We wanted to know if the baseline level of reservoir had an impact on the efficacy of this treatment. And as it turns out, we see a significant decay in the reservoir from weeks 5 to 12 in participants with a high baseline reservoir. So we took this decay rate and made a model. And what this model shows us, it predicts a near total clearance of the peripheral reservoir in about 2.86 years. And we acknowledge that this model is limited due to estimating the peripheral reservoir and not sanctuary sites, and that continued use of ruxolitinib beyond 5 weeks would likely alter this decay trajectory. In conjunction with this reservoir analysis, we did a massive biomarker analysis, and this really had 3 stages. So the first, we wanted to know what biomarkers did this treatment alter. The second is, was the alteration different from the control group? And whenever we look at the expression profile of those altered biomarkers, what we see in the blue is the treatment group, and in the red is the control group. And you can see that that expression profile is nearly completely flipped. The third round of analysis we did with biomarkers, we wanted to ask what biomarkers are associated with the reservoir decay. And for this, instead of looking and comparing between the 2 arms of the study, treatment or control, we actually looked at everyone in the study, and we looked at those who experienced growth in their reservoir versus those who experienced decay in their reservoir. And similarly, what we see is the expression profile of biomarkers between these 2. In the decay group versus the growth group, it's almost completely opposite. And so whenever we overlap all 3 rounds of this analysis, what this gives us is a very niche biomarker list where we're looking at markers that were altered by the treatment, different from the control, and associated with reservoir decay. And this gave us a list of 4 markers within various cell subtypes. So the first being BCL2 Ki67 expression in naive CD4 positive T cells. And this indicates productively infected cells with an increased lifespan. And so what we see with our treatment group in the blue here is that by week 5, it is downregulated from control. The second is CD127 expression in terminally differentiated CD8 positive T cells. This indicates a contained viral load, and we start to see this trend as an increased expression from control by week 5, but by week 12 we see that it is significantly decreased from control. And we think this is a potential compensatory effect that may change if the length of treatment was longer. pSTAT5 expression in monocytes, this indicates activation of the JAK-STAT pathway and is an upstream factor of several HIV persistence factors. And what we see here is a decay, a downregulation in expression of pSTAT5 by week 12 in the study. And then the last marker we have here is IL-10 in the plasma, and the interpretation of this one really just depends on the context you're discussing it with HIV, but our observation with this is that it was trending towards significance above control by week 5. And so of those 4 markers I just showed you, 2 are also shown to be predictors of reservoir decay. And so what we found was that baseline expression for BCL2 Ki67, if it's a lower expression, those participants were more likely to experience reservoir decay. And the second is looking at pSTAT5, and we found that a baseline higher expression of pSTAT5, those participants were more likely to experience reservoir decay. And what we know is that in our treatment, it reduced expression for both of these markers. So in conclusion, we have demonstrated that ruxolitinib treatment results in significant decay of the HIV-1 reservoir in those with a high baseline reservoir, and that our model predicts a near total clearance of the peripheral reservoir in 2.86 years. Many of the biomarkers we saw to be significantly altered were associated with things like viral persistence and immune dysregulation. And then I discussed those 4 markers with you that we found to be altered by our treatment, different from control, and associated with reservoir decay, 2 of which were also predictors of reservoir decay here. And so our main conclusion is that ruxolitinib, a JAK1/2 selective inhibitor, administered for 5 weeks in this study is a proof of principle that demonstrates this drug class can reverse immune dysfunction and also significantly decay the reservoir in a subset of people with HIV. And we think that longer duration of studies with this drug class are warranted and actually, in fact, underway. And I would encourage you all, if you were interested in this, to seek out the oral abstract by Asirase Siran at this conference, in which he had an intervention of Ruxolitinib, the same drug here, in an ATI case study for much longer than the 5 weeks that we observed in the study. And so with that, I would really like to thank the organizers of IAS 2023 for the opportunity to speak, and the Towards and HIV Cure team. They've been amazing, as well as my thesis advisor, Christina Gavagnano, and my colleagues and all of the funding entities here. Thank you so much.
So we have the general discussion. All presentations are open to talk and question or comments. So maybe for the latest presentation with the JAK2/1/2 inhibitor, did you haven't seen lymphopenia, cytopenia?
Monica?
I think Monica. Yeah, sorry to bother you. So no lymphopenia, cytopenia, because you block BCL2, so you may have less cells. So can you comment on absolute number of cells?
On the absolute number of cells, off the top of my head, I cannot. But if the concern is about blocking BCL2 and potentially that having some type of proliferative effect, we actually know that the JAK inhibitors do inhibit proliferation just kind of as like a side effect. But that has not been shown to have any type of severe side effects in humans that it's been approved to treat. The BCL2 really, from our perspective and our body of work that we know, is upregulated on reservoir cells. And so the fact that we see a reduction in that really, I think, correlates with this, this drop in the decay in the reservoir. Because if you're getting rid of BCL2, which is a lifespan marker for the reservoir cells, then it makes sense that you see this decay.
Okay, question here.
James McMahon from Melbourne. Great talk. Great to see so many clinical studies presented. Maybe Beatrice and Julie, I was thinking about this and some of the data, Beatrice, you presented, identifying a subpopulation that respond and have a better time in remission. And then it makes me think about BNAB sensitivity screening. We're getting more and more stringent with that. We've got other clinical factors, reservoir size, or early treatment. So it's a question about these studies, all of these trials. Should we be going, and get your comments about, down a path of a more selected population, around BNAB sensitivity and pre-responding to vaccines, which is not as generalizable to everyone, to generate a signal, or should we be altering interventions to encompass a broader population of people with HIV?
Hi, yeah. I think that's a great question. I think first we need to have the signal, and sometimes to get in that signal we need to shorten and reduce the population and try to see who can respond better and try to optimize that. And once we have that signal validated in those populations, then maybe we can build on that. So, I mean, of course, we want to cure for everyone, for all subtypes and for all type of people with HIV, but I think we are still far away from that. And I think the best that we can do now is to try to test and understand who is responding to the strategies that we are doing that. And when designing the combination trials, try to build on that and make that the next agent that you put in the combination is really going to enhance what you saw before, because if not, it's like we are diverting the signal. So yeah, but that's a difficult thing to do. But I mean, great question.
Yeah, and I think my answer would be both, right? And it depends on what population you're trying to address. So for when you have a known cohort of individuals that's treated and suppressed and you're going to do an ATI, I think that it makes a lot of sense to do as much pre-screening as you can. And I would say, for our next study, we're actually doing TZMBL screening for to confirm BNAB susceptibility in an ATI, and also excluding favorable HLA types, right? Because we've characterized these individuals in these cohorts very well. Absolutely, you want to select the optimal group of folks when you can. On the flip side, there is the immunotherapy during early infection at the time of ART initiation. A lot of times, you're not going to be able to, pre-screen people in that setting. And there where you talk about, adjusting your therapies to the patient population, that's exactly what I think you have to do. And I just think, what we thought in our case about what we would expect in terms of the in vivo, susceptibility to VRC01 years ago, is obviously very different than what we found out in not just my study, but many others. And so I think that, ensuring that the panels that are used to assess, global viral neutralization susceptibility always have to be updated. They need to be representative of current circulating strains, so that we're going to pursue this, I think, very promising strategy of intervening while people are relatively recently diagnosed and still have viremia. We're going into that with kind of choosing the best interventions, monoclonal antibodies, for example, based on the best possible information that we can have to inform that decision. So thanks so much. I think it's a great question.
So yes, we have time for only one more question, so sorry.
Okay, I'll be brief. One on the humans and the other on the monkeys. So for Dr. Beatrice, I think the presentation was really interesting, and if I understood correctly, it looked like you had the pre-HTI responses, but many of those seem to contract post the red circle, the red proportion sort of diminished. So I'm wondering if you have looked at that evolution of that HTI response with time, and whether or not it correlates at all with sort of slower viral rebound. So how critical is that HTI response, and how is it affected over time? And then similarly in the monkeys, so Afam, it was nice to see your data, but wondering if you had sufficient T-cell responses with the 9 days, initiating ART 9 days later, and whether you're doing any studies with vaccines in addition to the antibody immunotherapies.
Sorry, Beatrice, we need a short answer, please. You can continue the discussion later.
Sorry. The pies, they don't show it's contracted. On the contrary, they show it's expanded. So HTI pre-existing response is around 10%, and after vaccination, we peak it around 80, and we end at ATI start around 50. So vaccination, what it does is to expanding our pre-existing response to HTI, and that correlates with the outcome.
It did diminish. So immediately post ATI, yes, but I thought later on the response diminished.
No, that's not the case, sorry.
So with this, we close this session. Yes.
Well, the answer is yes.
Ricardo, don't kill me. We will now have a lunch break for 1 hour and 10 minutes. There will be no lunch provided by the organizer, but there is a lot of cafes and restaurants in the surrounding area. As Sharon said earlier, enjoy the Brisbane food, but please be back at 1:45 so can stick with the scheduled program. Thank you very much.